Many of you already know Rob, or maybe all of you already know Rob. But I'll just run through his academic history real quick for those of you who might not have met him yet. Rob grew up in Illinois, did his undergraduate at Syracuse, his master's at Indiana University, came here for his PhD with Peggy in my lab, and worked on mycorrhizal fungi. And he's not going to talk about his dissertation work here. It's really interesting though. But then he had a postdoc with Amy Burgin's lab in the Ames project, also taught at Haskell University as part of that postdoc. And now he's at the ESIIL, which, what does that ESIIL stand for?
It's the Environmental Science Inclusion Lab. There's another I. I don't remember the other I's for, but it's the Inclusion and Equity Lab, I think.
At the University of Colorado at Boulder. Yeah. And so so we're very happy to have Rob back visiting. He's Rob and Camille have been working in the molecular lab doing trying to get DNA out of AMF spores.
We will succeed.
And so I think today Rob is going to talk about merging the work on the microbial patterns with the Ames project with the microbial patterns that have come out of the MAPS terrestrial sampling. So Rob.
Thanks so much, Jim. Thanks everyone for coming. I'm going to hide the video real quick so people don't have to. Oh, did I stop sharing?
You're fine on here.
Oh, okay, I just blew this up. Okay. I did. I'm unmuted now. I'm muted now. Good. All right. Cool. We're all set. Thanks everyone for coming. So yeah, I'm going to be talking a little bit about some of the work I've been doing in my postdocs. And I totally planned this, and isn't an accident, but a lot of my postdoc work has this running theme of like spatial patterns of microbial communities. So I'm going to we're trying to understand the spatial patterns of microbial communities, scaling from projects at a local scale in Kansas to statewide, to now trying to do some work looking at the continental US and scaling up some of my questions. And I had to put in the comment, maybe everything isn't everywhere, totally in response to a reviewer comment. But yes, trying to understand these spatial patterns and like what's driving them. Is it always environmental filtering, or are there other more important dynamics happening? Alrighty. So I'm going to break this up into 3 scales. Work I did at on the Ames project. We're looking at the Ames project spans larger than this, but I'm going to be talking about work that we did at Konza, looking at the South Fork of Kings Creek and the spatial patterns of the microbial community in this intermittent stream. So a stream that's drying over the season, and its network connectivity is changing. All kinds of fun things are changing about that network. Then we're going to go into looking at just AM fungal communities, and I'm sure you've all heard AM fungi before, but I'll do a quick brief introduction at the time about what those critters are, and looking at their distribution across depth and the precipitation gradient that exists in Kansas. And then I'll briefly talk about what I'm doing now at ESIIL and where my research is going. So starting, we're going to talk about these patterns of microbial abundance and diversity relating to I'm calling it discontinuity here, but basically as the stream is drying, all kinds of stream characteristics are changing. There are breaks in the system, and there's it becomes a very complex network over time. And overall question is, how does that change in connectivity affect the microbial communities? So why do we care about microbes and streams? Microorganisms are the backbone of the food web in most streams. And they've, it's these trophic interactions have been, studied a long time, but also just microbes are great because they comprise the majority of the biodiversity on the planet, and they're an important group in and of themselves to understand. So trying to understand this project, I started with like the very basics, right? Like trying to understand what's going to be structuring the communities in these streams, all the way back to the river continuum concept. Trying to understand, you know, in this case, it's usually considered in the base of stream order, right? So, but this is an intermittent stream, so it's really like a 1 and 2 order stream. So not necessarily stream order, but expanding that out. We're talking about, you know, there are these kind of these communities kind of change in structure and their function as you're moving throughout the river. Low order, you know, headwater streams, you know, there's a lot of terrestrial input, a lot of leaf litter, lots of other kinds of, thank you, terrestrial input. So we expect these to be dominated by heterotrophs and just a lot of influence from that terrestrial inputs. And as you move into higher order streams, you get wider streams, you get more in-stream processing, and so like you end up with these more autotrophic systems. And you know, there's still influence from the terrestrial environment, but in relative proportions, maybe they're less important than it would be in the headwaters, right? So that's where I started. As a kind of like dueling hypothesis, I leaned on this residence time hypothesis. And this is all about this kind of in-stream processing. So the idea in this framework is as you get more downstream, organisms have had more time in the water column. And it's that selection pressure, right? Everything's everywhere and the environment selects. So as things spend more time in the stream, you're losing taxa that came in from the terrestrial environment, and you're only retaining your aquatic specialists. So overall diversity goes down the longer you've been in the stream. So these two kind of competing, sometimes they work together, but you know, sometimes they're competing frameworks. Kind of how I started structuring my thought process early on in this project. So how are we going to look at this? Well, first, skipping a step, why intermittent streams? Why even look at intermittent streams? So just general, very important slide for the Ames people. Why are we looking at intermittent streams? It's because they're very prevalent, like 50%, I think that's right. I didn't have it on the slide, but I think it's 50% of the stream networks in the US are intermittent. They dry regularly, either seasonally or maybe at most every 1 to 2 years, but they dry regularly. And that number is going up with climate change. With climate change, we're getting more and more stream miles that are intermittent. They dry periodically, and we don't really have figured out the methodology for understanding the systems. A lot of the classic methodology for understanding streams assumes that there's always flow. So there's a lot of work that still needs to be done understanding how do you study a stream where the thing goes dry part of the year. So research question, what is the impact of this hydrological and the habitat connectivity, right? You're fragmenting the system as things dry on the microbial communities. That was our overarching question. So we had some hypotheses. Higher hydrological connectivity will cause more times of bacteria and fungi to be present at one location, so higher alpha diversity, right? You're just having more things in the system, so you're gonna have higher alpha diversity. And they'll deliver more nutrients, right? So you're getting more stream flow, nutrients are flushing in, you're gonna have higher abundances. That was our starting hypothesis. We also had some hypotheses, so the sampling design that I haven't talked about yet in Ames, we specifically sampled 4 substrates. So we started water column, we sampled biofilms, lots of scrubbing rocks. We also took sediment samples and leaf pack samples. So when I say leaves, I mean leaf packs in the stream. All right, so now the slide I thought was coming. How do we quantify discontinuity? So this was a huge discussion. A lot of the people on the project have thought about this. We've all come up with similar but different ways to quantify the discontinuity. I'm going to talk about the instrumentation we use and then ultimately what metric I used. So the instrumentation we used are the stream temperature, intermittency, and connectivity sensors, stick sensors. They indicate presence absence, so the connectivity sensors are the main thing we're using. You can use temperature for corrections as well, but mainly connectivity is what we're using. And then we put 50 stick sensors across the network. We could do that because these are actually really cheap. Like they're sub $100 per sensor, so you can kind of put quite a few out of them out there. They're relatively sturdy. We definitely have had a few burn and some fires, but for the most part, they're sturdy. And we have them set to record every 15 minutes. So high temporal data density with these sensors as well. So continually monitoring for 15 minutes, 50 sites across this relatively small stream section. So pretty high data density. All right, but we have presence absence of water throughout the network. How do we turn that into useful metrics? So again, a couple different approaches that we're using in the project. This is what I did. So it's called communication distance. It's a refinement of the stream length duration curve that Potter and Durgato came out with a couple of years ago. But it's basically a weighting of stream lengths based on how probable you think the stream is actually, there's water in the channel. So minimum values are the physical stream length between two sensors, and then it kind of grows as there's less and less likely to be water in the channel. So you can kind of interpret this as like a resistance to transport between two nodes in the network due to water and water flow in the channel. So that's the metric I used. And with that, we will take these 50 nodes of presence absence and turn them into these kind of like numeric values between each site. And we use that as our metric. All righty, so just real quick, just putting up there the actual model I ran. So these are alpha diversity is our metric in this study, and then we're looking at this this communication distance. In this particular, I didn't have time to update these slides. I've refined this slightly since I made these slides, but in this particular analysis, we're summing up this communication distance to the node. So this is kind of a metric of how much it grows as there's more discontinuity in the upstream network, right? As there's more breaks in the upstream network, you get a higher number here. So that's how we did it in this analysis. Also looked at stream order in this analysis because I was trying to compare it to the traditional, river continuum concept. I do realize it's a little limited in this case because it is a pretty much all a headwater stream. We go from 1 to 3, but I was attempting to kind of bring in that classic framework as well. And then flow state, because we just learned really quickly, wet or dry makes the biggest difference. So we definitely had to include that in the analysis. And then we did separate models for each microhabitat. So we don't have flow state when we're looking at the water because we couldn't sample water when the stream is dry. So that's what we did. Looking quickly at results, I grayed out the things that weren't significant. So what we had is significant alpha diversity by stream order effect in our leaf packs. And since this is actually one of the ones where it lines up with one of our predictions, leaf packs, we saw the greatest diversity in the headwaters. And so as we're getting further and further down the stream, we're actually losing taxa. And this would be consistent with this hypothesis of like, as you're spending more time in the water column, you're losing extra taxa with this there because it got flushed in from the surrounding environment. Now moving past stream order, now we're looking at that CCD metric. So this is a more continuous metric. Again, only thing that was significant was the leaf packs. So as we're getting more discontinuity in the network, our alpha diversity is going up. And I think this also makes sense, right? Like you're getting more patch heterogeneity. The overall alpha diversity might be going up. That's really more of a beta diversity trend though. So to be honest, still not fully sure I understand what's going on with alpha diversity here. I can kind of make an explanation here, but I gotta admit, I'm still a little puzzled about alpha diversity going up as we're getting more discontinuity in the stream. And then finally, flow state really mattered. Oh, and I missed a step. These are all 18S, I believe, results. So this is all the eukaryotes in the stream, right? So yeah, it mattered a whole lot if it was dry or flowing. Truthfully, we kind of expected the opposite result. So when we started, oh no, I'm sorry. I'll get to that counterintuitive result later. This makes sense. The flowing states had higher alpha diversity. So this is what we expected wet or dry to be doing for us. So I think moving on now is, oh wait, hold on. All right, now we're at 16S. Okay, so now we're at bacterial. Okay, so in the bacterial communities, stream order mattered only for the biofilms, and then it starts going up as you get to higher order stream orders. The way we interpreted this, because it is reversed from the leaf packs, is that when we're talking about bacteria, that's also including autotrophs. So this could be a sign that we're actually increasing the number of autotrophic diversity as you're getting into more open stream networks. It's still on my list to break these things out by taxa, and I think that'd be really important to fully understand this, like actually break things out from autotrophs to heterotrophs. Right now, they're convoluted in just one alpha diversity metric, but it was interesting to see. Like, so we are seeing stream order as a significant predictor of the biofilms. And again, the biofilms are generally a lot of autotrophic organisms. So that kind of leads us into that understanding of that result. And then finally, or not finally, two more. Sediment. In the sediment community, we see a decrease in alpha diversity with our kind of discontinuity metric. So as the system is getting larger disconnections upstream, we're seeing a loss of alpha diversity. Oh my God, I'm so sorry. I am messing up. This is not alpha diversity. This is abundances. So same idea for the alpha, for the, to go back here. This is abundances, not alpha diversity. That's an important distinction, but it's the same idea. Autotrophs are dominant in the biofilms, and that's why we think they're increasing as you get more open water. Here, again, abundances are going down with the sediments, but it's going down when you have more discontinuity. And honestly, this one puzzles me. I'm not quite sure the explanation. If anyone has ideas, I would love to hear it. But it is the significant result when we looked at abundances in our 16S communities. And then this was the weirdo. This was the weirdo I was alluding to earlier. So we got abundances here of the bacterial community. Flowing, we have lower abundances than the dry. And we didn't really expect that. And so what we think is happening here, one of those things like, really wish we put out O2 sensors in the sediments, because we think it's an oxygen story, but we have no way to prove it. But when you are flowing, those sediments are likely going anoxic, and that's going to drive down the overall abundances. Again, we're assuming that we didn't actually put O2 sensors out there. So we can't say for sure that's what's driving the abundances to go down, but it is what we detected. Abundances went down when there was water present in the sediments. Again, no other significant patterns. So that is our explanation for that. It is interesting. Okay, so I lied. I didn't actually present the fungal data. I'm sorry. So the conclusions we have are that the river continuum concept, the residence time hypothesis, just general connectivity measurements. It's not a clean story. Different substrates were behaving differently, and so that's important to consider, especially when you're dealing with these intermittent streams. The substrates can behave very differently from each other when things are dried down. So that's something to keep in mind, that we do kind of need to look at these different compartments and don't necessarily expect the same kind of patterns within these compartments. And then, binary wet dry was very, very important. We think that's an oxygen story, especially in the sediments, and not necessarily in the way we intuited that it would work. So that's what I'm going to pause there, and we're going to switch now. So that's what I found looking at these microbial communities localized to a single stream network in Kansas. What I want to talk about now is a different project where we kind of are looking at microbial community spatial patterns, but at a much larger scale. So this is the MAPS project. These are terrestrial sites across the entirety of Kansas. And then I will scoot on ahead. And for example, we have samples from right here at the field station. Also had some samples at Konza, and we had many more sites as well. I'm going to back up again. So in this experiment, I'm not looking at total microbial community dynamics. I'm an AMF guy. Jim talked about this. I did my PhD looking at arbuscular mycorrhizal fungi. So we're only looking at AMF in this analysis. Why are we looking at AMF? Because they're fantastic organisms. That's why we're looking at AMF. There are fungal partners that associate with most plant species. They've been known, they have been demonstrated to improve resource uptake of their host plants, particularly phosphorus. They also have been shown to have benefits for like water uptake and other benefits as well. And we do have plenty of studies at this point that show that AM fungal diversity can affect their host plant community function. All right, now I'm supposed to have a slide here to kind of talk about the overall design first, but we'll go over the design elements of MAPS in sequence. So MAPS had two important dimensions of the spatial component, looking at these different communities. One was depth. Now, why did we want to understand these communities at depth? Well, these are all grassland systems, and depth is very important, right? So these are very deep rooting plants. These are root-associated microbes. So we don't really have a great understanding what's happening under the first 15 centimeters. And it is probably important for this group, right? There are roots down there. These AMF associate, they grow into plant roots, and that's how they facilitate the mutualism. So a better understanding of what's happening at these deeper soils with these communities was something we thought was really important. There's also a land use history component to it, and it interacts with depth, which is, of course, people here who work with land institute will know. Traditional agricultural crops tend to be a lot shallower rooted than a lot of the prairie species out there. So we got switchgrass, annual wheat. You can see the difference in the root depths. So it also maps on to like these very deep rooted plants. They're pretty different than a lot of the typical ag species that we have. So we wanted to understand the land use patterns as well. Prairies have been plowed for agricultural use. It's very disruptive to soil communities. So we definitely wanted to include that in the MAPS analysis. And then again, one of the big things MAPS was set up to study was the fact that we have this beautiful precipitation gradient across the state, right? We have this drier climate in the western part of the state and a much wetter climate on the eastern part of the state. So we were sampling across Kansas in order to capture this change of precipitation that you get from the eastern part of the state to the western part of the state. And that, of course, I wrote this for people who don't live in Kansas, but forgive me, that's very important because you get an ecosystem change throughout this precipitation as well, right? You get the shortgrass prairie at the very far end, western end of Kansas, where you still have tallgrass prairie here in the eastern part. So like there's also a community shift as you go across this precipitation gradient. So sampling across Kansas, we're sampling through depth, and we're sampling across different land use histories. Agricultural systems, post-agricultural systems, so fields that basically have been fallow for at least 10 years, and native prairie that's never been tilled. Or I think that's true, always never been tilled, yes. Alrighty, so that's the experimental design. Again, gave this for more general talk before, so apologize, but I'm gonna go over alpha and beta diversity real quick. So alpha diversity is one of the things we're gonna look at. It's what we looked at in the first part of the talk. You know, high alpha diversity, everything is more or less evenly distributed. You don't have single species that dominate the overall population or overall community. Where low alpha diversity, you have individuals that are more dominant. There's more of those individuals in the overall. Opposed to beta diversity, now we're looking at the heterogeneity of the entire system. So in a high beta diversity system, each individual, part of the community may be very, very different. In a low beta diversity system, any particular part of the community is going to be very similar to each other. Right, so we're going to look at both aspects of diversity in this study. Just to recap the study design, this should have been first. 12 sites across the precipitation gradient. We sampled across 2018, 2019, and 2021. We crossed those 3 land use histories, and we sampled at different depths. So 5, 15, all the way up to 150. I don't need to read them all out for you. So we went pretty deep. We used the Giddings. If you guys have seen that thing, it's a monster machine. So we went pretty deep with these sampling. All right. So what did we find? I am going to present these tables just to show you that I did a permanova. We don't need to dwell on this real quick. But the beta diversity is important to look at. We got a lot of significant drivers of beta diversity out in this study. So land use history was important, depth was important, and precipitation was important. We also had some interesting interactions that we will talk briefly about. I don't want to dwell too much on the interactions, but we did find a land use by depth interaction and a land use by precipitation. We'll look at graphs of these and break them down a little bit more. With the alpha diversity, it wasn't as many drivers, but we did see a driver of alpha diversity with depth, and we did see land use interacting with depth. The only time precipitation was important for alpha diversity ended up being in complicated 3-way interaction, which I'm sorry, but I will walk through and we'll get through it. But it's a 3-way interaction where depth ended up being important for alpha diversity. So let's kind of step through these now with some graphs so we can better understand exactly what's going on. So with beta diversity, again, land use history was an important predictor on its own. And I was so happy when I saw this plot. You guys have no idea. It made sense. It was what we predicted. So ag systems, post-ag systems are in the middle, and then native systems. So we kind of ordered in them in the idea that post-ag systems would be an intermediate between whatever's happening between the ag systems and the native systems. And sure enough, that's what we found. The lowest beta diversity was in the ag systems. So you take multiple samples in an ag system, they were more similar to each other. And then the native systems had the highest beta diversity. And then post-ag happened to be in the middle. This was so beautiful. Maps worked. We saw what we expected to see between the land use histories. I was super jazzed to see this plot. Looking at depth, didn't really see an interesting. The line's pretty flat for beta diversity. Alpha diversity, we did see something kind of cool. So depth, you see alpha diversity going down with depth. Interesting. Honestly, I expected to see the beta diversity not be flat. So I expected the deep soils to be more similar than topsoils. We didn't really see that. Very strange. We didn't see it, but we did see that overall we had more dominant species at the lower soils. I think that's evidence there might be some cool specialists down there doing something interesting. We haven't quite figured out who those might be yet. Again, need to do all the taxonomic, like figuring out who exactly is the players here to really get at the details. It looks like we do have some deep soil specialists in the AMF community, which is very exciting. I'm super jazzed. And then looking at precipitation, again, I'm sorry, I will walk everyone through the three-way interaction. Beta diversity is a simpler story. As you get wetter, you lose beta diversity. Not what I was expecting, but that is what we found. So like the wetter sites were more similar to each other than the dry sites. There was more heterogeneity in the drier sites. It's not that it doesn't make sense. I just kind of in my head intuited that the wetter sites would be have higher beta diversity, but it turns out the drier sites in Kansas have a higher beta diversity. They're more dissimilar from each other. Interesting. This guy. We'll talk about the three-way. The way I interpret this three-way is that I split it up for the graph here. So we have our mesic sites, our wettest sites. Split this basically our sites in half. So we have the wettest half and the driest half. Basically, everything's a little bit stronger in the wettest half, and we have an interesting thing that's not happening in our post-ag sites, in the dry sites, that is happening in the wet sites. And that's basically the ag sites. If there's any trend at all, it's slightly positive with depth. So basically, our alpha diversity, I would interpret this as our alpha diversity is slightly lower in the top soil. And if you're dealing with a system that's constantly plowed, that might make sense. You're knocking back species because you're constantly plowing the fields. Again, it's a weak trend. I think it's significant. It might be significant in this plot. I don't think it's significant in this plot. But yeah, in the post-ags, the dry sites, just a flat line. The wet sites, though, look like our native sites in that as you get deeper, you're losing, again, this is similar to what we saw in the effect of depth. And that exists very strongly in the native sites. It doesn't exist as strongly in the ag sites. And then the post-ags have this very interesting difference between our wet sites and our dry sites. This, I would love to hear anyone's ideas about why that's happening, but it is interesting. And again, this three-way interaction was the only time precipitation was significant with alpha diversity. And then finally, we have some interesting interactions additionally to this plot with land use history. So both the beta diversity and alpha diversity, when you're dealing with beta diversity, it's precipitation, and with alpha diversity, it's depth. Both interact with land use history. Again, the ag systems seem to have the weakest relationship with precipitation. Irrigation could very easily explain that. And then the alpha diversity, the depth. Again, I don't think this is significant. It's a positive trend, but I don't think that's significant. These two are, and they're both similar to each other. Post-ag and native both have an even stronger relationship with depth than the ag systems do. So, in conclusion, depth mattered. It mattered there were different communities, and the communities at depth had more dominant species. Precipitation mattered. The dry sites were more heterogeneous. Cool. Not what I expected, but cool. And differences at depth are greater at the wet sites, which is interesting. And then land use history was also important. Agricultural sites had lower beta diversity, they were less heterogeneous. And agriculture was disrupting both trends in precipitation and depth with beta and alpha diversity. So I'm super excited about these results. People who have worked on maps knows that project has taken a long to get all the analysis done, and we're starting to see some really cool patterns from all the work that was done on maps. But this whetted my appetite, and this is actually part of the work I'm doing now at ESIIL, was starting with this data set to include it into a larger data set. So what am I doing next? Well, I'm taking some of that data set and I'm combining it with a larger data set that's global AM fungi. You guys people might be aware of that site. And we're trying to now take this AMF data for the map stuff. I'm only taking the top 5 centimeters. But to take this data and now try to understand how AMF are distributed across. I have limited myself to the contiguous US because I looked at the data and realized we don't have a lot of the rest of the world. So looking at just the US and trying to understand not only how AMF are distributed, but because they're an obligate symbiont, I'm trying to do this alongside modeling their host plants. Maybe with a joint species distribution model, maybe with something more exotic. I'm still figuring the mechanics of that out, but got a lot of the data pulled together, so that's fun. And yeah, so we're doing modeling effort with curated studies of the LSU gene, specifically. If you guys know Bever Lab, we love the LSU gene for AMF. Makes bioinformatics really, really hard, but we love that gene, so I'm trying to use that. And then probably GBIF for the plant data. So this is the next thing I'm working on. I've run my first models this week. I got really excited and started running some models, and they're terrible, so I can't show them to you yet. But actually starting to get into the actual data analysis, which is exciting. So this is what I'm working on currently at ESIIL. And with that, I didn't put a question slide in, but I'm happy to take any questions. I'll repeat that. So could precipitation be homogenizing in the community? And that is why we're getting lower beta diversity. It's possible. I hadn't considered that, but definitely could be. Like there's just more organisms moving through water in the wetter sites. I hadn't considered that. That's a very good explanation. With the MAPS data, I don't know if we could really be sure one way or the other, but definitely that's a very valid explanation for why we were seeing that pattern, and I hadn't considered it. So definitely could be very interesting. Yes. Yeah, so am I seeing similar patterns at the wet and dry terrestrial sites and the wet and dry aquatic sites? Honestly, this is the first time I've smashed these two projects together, but it is a very good question. I would have to dig into it more. I think it's very valid to try to look into both these data sets in that way. I don't have an answer for that yet. Yes. Yeah, so I'll repeat for Zoomland. So the comment was, with specifically the wet dry, I think this was the wet dry and the sediments for abundances. One of the explanations could be that because this is a stream, it's not that dry, that these are wet alchemated microbes, and maybe they're just getting kicked back because they're not really adapted for the dry site. Yes, that could be an explanation, and because we don't have oxygen sensors, it's really hard to say. So yeah, there's an interesting pattern there. It is hard to disentangle. We really wish in hindsight that we put oxygen sensors in the sediments, but you design these experiments and then you realize after the fact all these things that you could have measured and didn't. So I agree. It is hard to disentangle those two, and I'm sure there could be other explanations as well. Yeah. It's supposed to be me. But I haven't gotten that far yet. Yeah. That's yes, exactly. No, it will be, and I will find the time. Yes. I would actually say, and this is why I put this continuity on the slide, but we've kind of changed the verbiage on this all the time. But yeah, it's a measure of connectivity, physical connectivity between the sites is the way I would interpret it, right? Surface flow, specifically. We have these all conversations about surface flow and subsurface flow and what we can and cannot measure. But yeah, surface flow hydrologic connectivity is the way I would actually interpret that. The way I used it in that particular analysis is kind of a discontinuity because it's kind of summing up the upstream, but it's not really perfectly tuned for that question. So it is a better metric of just like hydrologic connectivity for sure. It would be a little bit sensitive to exactly where the spring was and where our sensors were. But ideally, yes, we would capture the spring. So we'd have a wet site that was spring fed. It wouldn't touch any of the other sensors around it. And that would end up being a very high value of communication distance because there's these big breaks. And so those breaks would inflate the overall distance, right? So we're weighting distances by how likely there is to be water between 2 points. That's an important point I didn't mention here. It's calculating between 2 nodes. So yeah, a spring-fed site would be wet, doesn't connect to anything else. That would have a very high communication distance. And then so we definitely had a handful of wet sites that were at the time of sampling. Something I didn't mention here is like this is looking at our biggest sampling event. There's a lot of seasonal data that we also collected for this project that still hasn't made it into these slides. And I'm really interested to see if we can get stronger signals when we actually incorporate the whole seasonal sampling that we did. But yeah, we would theoretically have a lot of sites at Konza because it's so spring fed and there's like really weird drying patterns out there. We have a wet site that's very disconnected and that would show up in the communication distance.
I think we're ephemeral. Ephemeral didn't make the cut.
I would let the hydrologist argue about whether or not we have ephemeral streams. I think in the Mountain West, so this is multi-state, and I'm only talking about Kansas and I'm only talking about Konza. Mountain West, some of them were arguing that their sites are actually more ephemeral, like snow melt and then they're gone. But I think in Kansas, it's safe to say what we had was intermittent streams at all of our sites. Yes, Laura. With the exception of wet dry, because we don't have any dry water sites. So otherwise the models were the same. I think I may have saw Connor twitch when someone mentioned flow rate. we have flow rate at the outlet now for this site. We didn't when I made these slides, but we only have it at the outlet, right? So our rest of our network was instrumented, which is water presence absence. So things like flow rate, it's hard to estimate that back throughout the network. Yeah. Yes, Connor. We didn't have enough pooled for the synoptic. When I include the seasonal, absolutely. Tara, you've been sitting on your hands. I'll be honest. So I played with it, which isn't the most scientific way of doing this, but I had a 30-year window and I calculated different averages. And just the one that had the most statistical predicting value was a 5-year average, actually. So I did play with like the same year, the previous year, the 30-year average. The 5-year average seemed to have the strongest predictive value. No, as far as I know, unless someone in Maps wants to shout out that that data existed, I don't know about it. I don't think so. I think that's everybody.